Hello, Ramesh, can you hear me? So we will wait for a few minutes so for others to join and then we shall start this session. So have you given your assignments, assignment 1 and 2? Okay, so how is it? So can you see the marks instantly or something? Okay, but what is the deadline for assignment 2? So deadline is over then. For assignment 2, the deadline is 10th August. Okay, okay. So you have submitted both. Okay, okay, okay. So then you after submitting, so you can't see how much you scored out of 10. Okay, okay, okay. So have you? Yes, say. Uh, yeah. So actually, there is no kind of model question because every year it is new things are sometimes new questions are made, sometimes they are not made, so they repeat the question, so they are not shared publicly. Okay, okay, so it is a pen and paper, so it is not MCQ or MSQ or what you are giving in assignments, it is not like that. So the professor will make the questions for the final exam, so it is descriptive. Yeah. Okay, so I will talk to the professor and I will see if there are old some question paper or something which if he shares, then I will share it with you. Okay. Hmm. Yes. Yeah, so how to solve the structures is there in this course in the later weeks. Yeah, so at first the professor is teaching the basics, then also the theory part. So because so when we are collecting it, we are collecting in the diffraction space, right? In the reciprocal lattice, we get the reciprocal points. So you solve using chrysalis, huh? Okay, means okay, okay. So where you will, so before solving, so the professor actually teaches us how to convert this diffraction space, these intensities into the electron densities to understand which is carbon, which is nitrogen. So it is transformed by something which is called as the Fourier transformation. So all this he will teach up to like week 6 or 7. Then after that the hands-on training and those will be there. Yeah, so you are in which institute? Oh, okay, so you're a faculty. Okay, okay, so we can start. It is 3 or 7. Okay, so let me share my screen. So basically, this is the outline of this course, the chemical crystallography course. So as you all know, so it has started on 25th July. So this is the third week, and so for every Saturday we have this live session, and this is the Google Meet link. So where we are having these sessions, and then so this is the swam portal of this course, and then for everyone, so when you have any question or something, you can post this here in the ask a question tab, and here when you post it, so the TA or the professor will be able to see the questions and they will answer accordingly. And then so now we welcome you to this to the third week and to the third live session of this course titled chemical crystallography. So till now we have learned about the absolute basics, and also like what is this X-ray diffraction and why do we study this X-ray diffraction. So we perform the X-ray diffraction experiment on crystals, primarily single crystals which have very small sizes, and then what we do, we shine x-rays upon them, and then there is the phenomena of diffraction. Then the diffracted intensities are collected on the detector, and then with the help of a computer and a lot of mathematics, the crystallographer solves this using, with the help of softwares, to get the 3D electron density map to understand the various positions and locations of the atoms, and thereby construct the molecule. So what is the most important advantage of this X-ray diffraction is to view atoms in the molecule which are separated by very small distances like 1 to 2 angstroms. And also we have, when we have many spectroscopic techniques like we have IR, we have mass, then we have mass spectrometry, then we have IR, also NMR. These are very useful tools for a chemist. However, this X-ray diffraction is very advantageous in the sense, suppose when we want to understand what are the bond angles. Suppose we have a carbon atom. Is there some sound coming? Can you please mute? When we have, suppose, a chiral center, and then where the carbon atom is attached to four different kind of atoms, and we want to know the absolute configuration, so whether it is R or S, then it is very difficult to understand it. So as I was saying, so it is very difficult to understand it from NMR, so that is why the single crystal X-ray diffraction is very much useful. There is some noise coming. Okay. Yeah, it's okay. Thank you. So that is why this single crystal X-ray diffraction is very useful, because we can get the precise information about bond lengths, bond angles, and also the absolute configuration, among many other things. So now when we, so in the first few lectures by professor, we have got the preliminary idea about symmetries, about point groups, and also about space groups. So we now know that there are 32 different point groups, and point groups are enough to define the symmetry of a molecule. However, when we go to the lattice, when we are talking about a crystal lattice, then in a crystal, these molecules or atoms or ions are arranged in a regular periodic three-dimensional pattern to which for theoretical purposes we consider as infinite. Mm, but we know that in the practical sense a crystal is finite. It has a definite dimension in length, breadth and thickness. However, so when we talk about it theoretically, we say that the atoms or ions or molecules, they are arranged in an indefinite three-dimensional array of points which are called as the lattice points. And when the molecules are arranged in the three-dimensional matter, then the most important symmetry in the crystal is the translational symmetry, because the molecules are related to, will have to be related to each other by translation. So this translation symmetry is the most important symmetry in the crystal. And the after that, the after this basic translational symmetry, the way the molecules or atoms are arranged give rise to the different space groups. So when we talk about a point group only defines the symmetry of a molecule, and then when these molecules are arranged in three-dimensional space, it gives rise to space groups. Now when we talk about space groups, there can be different types of space groups. So from point group, from the 32 different point groups, it gives rise to, we know, 230 different space groups. So among these, there are some which are centrosymmetric, some which are non-centrosymmetric, and some which are chiral. So I want you to answer. So what exactly are centrosymmetric space groups and non-centrosymmetric space groups? So chiral we will talk later. So what are the centro and non-centrosymmetric space groups? So can anyone say, please? So there are two kinds of so before space groups we have talked about point groups, right? So there are centrosymmetric point groups and non-centrosymmetric point groups. So what is the difference between them? No, twofold is not centrosymmetry. The basic, so from we talk when we talk about crystal lattices, we define it on the basis of symmetries. So the inversion symmetry is the operation based on which we define point groups and space groups into centrosymmetric and non-centrosymmetric. If there is an inversion center, then it is centrosymmetric. If there is no inversion center, then it is non-centrosymmetric. So for example, if we one second. So we know the CO2 molecule, right? So CO, pardon my writing please. So CO2 molecule, and then we have water molecule, right? So if we draw it, the CO2 molecule, this is the carbon, one side is oxygen, another side is oxygen. So this falls in the category of a centrosymmetric molecule. Why? Because this point, If we consider the atoms as spheres, then this is a center of symmetry because this side and this side is equivalent. However, if we draw water molecule, so it has a bent structure, right? V-shaped. Water is a V-shaped molecule. Now, if we consider the center like this, then one side we have hydrogen. Then what is the opposite side of this hydrogen is somewhere here, and here we do not have an identical atom. Hence, this is a non-centrosymmetric molecule. So this inversion center, which is defined as I. So this inversion center determines whether a point group or a space group will be centrosymmetric or non-centrosymmetric. And after we come to after we divide the space groups into centrosymmetric and non-centrosymmetric, the chiral space group is a subdivision of the non-centrosymmetric space groups. Essentially, a chiral space group is one which basically has a chiral center. So if there is a chiral center in the molecule, it will crystallize in a chiral space group. So are there any questions? Yeah. So can you give me the example of a chiral molecule? Hello, can you hear me? Yeah, so I was asking, so can you give me the example of a chiral molecule? So a molecule which has a stereo center. Can you tell me one chiral molecule? So a molecule which has a stereo center. Any molecule. So just like we have benzene, we have benzoic acid. So simple molecules like that which have a chiral center. Anyone? Two chloromethoxy. Yeah, so we can make a simple molecule like this, right? So yeah, so one side is nitrogen. Suppose this. Yeah, so before going to sulfur phosphorus, we can think of simple molecules, right? So what about amino acids? So this is methyl. Methyl, okay. This is methyl. So this is a stereo center, right? Hello. I can't hear. So some sound. Glutamic acid. Yeah, so it's okay. So anyway, so the exact molecule is not important. I'm talking. So let us talk about this molecule. So this is a chiral center. The carbons it is attached to benzene ring on one side. There is methyl group on one side. One side there is acid. One side is amine. So there are four different groups. This is a chiral center. Now when if we get crystals of this molecule, it will belong to a chiral space group. Why? Because this is a chiral center. chiral molecules, so when we talk about drug molecules, so drug molecules you usually have a chiral center. So there are big molecules and like levofloxacin and molecules like ofloxacin is the racemic, like levofloxacin. Then we have ciprofloxacin. So all they have chiral centers and they always crystallize in the chiral space groups. So what exactly is the difference between the centrosymmetric, non-centrosymmetric, and chiral space groups? So at first, let me give you some hints. So in crystal systems, so these are the common symmetry elements. There is translation, as I say. So translation is the basic symmetry element. We have rotational axis, we have mirror planes, and we have center of inversion. So, based on these symmetry elements, can you tell me what are the differences between centrosymmetric and non-centrosymmetric and chiral space groups? So, we know what is translation, right? So, if we have a molecule here, then it will translate in the x direction and also in the y direction. So this is the basic translational symmetry. Then we have rotational axes like two-fold, four-fold. Then we have the mirror planes like M. And then we have the center of inversion, which is I. So based on these symmetry elements, can you tell me what are the differences between the centrosymmetric and non-centrosymmetric space groups? Hmm. Yes. So yeah, so then let me explain the rest three. So first let us talk about translational symmetry. Translational symmetry is the basic symmetry. So centrosymmetric, non-centrosymmetric, and chiral, all the space group will have the translational symmetry. All of them can have the translational. All of them will have the translational symmetry. Then we come to the rotational axis, like two-fold, three-fold, four-fold, and six-fold. Right. So when we talk about rotational axis, then centrosymmetric space groups, non-centrosymmetric and chiral space groups, all of them can have rotational axis. They may have or may not have, but the important thing is that they can have. It will not hamper the symmetry. But what will hamper the symmetry is the mirror plane. So mirror planes essentially means reflection. Right. So, if we have a mirror, so can you tell me what will be the problem if we have a mirror plane in a chiral space group? Yeah, exactly. It will not be chiral because if we are crystallizing a pure enantiomer, suppose R, the capital R. So if we are crystallizing only the pure R enantiomer and we get the crystals of it, but if there is a mirror symmetry in the crystal structure, it will generate the S enantiomer. So it will contradict the basic, what we are starting with. Hence, mirror planes can be in centrosymmetric and non-centrosymmetric space groups, but chiral space groups will not contain the mirror planes. And then the last symmetry element is the center of inversion. Sorry, so only centrosymmetric space groups will contain the center of inversion. The non-centrosymmetric and chiral space groups will not contain the center of inversion. Now for chiral space group it is the same thing. So the center of inversion will generate the other enantiomer. So these are the differences between the different space groups. So now the yes. Yeah, because when we have, hmm, oh, yes, see, uh, when we talk about molecular symmetry, we have two kinds of rotations, proper and improper, right? So the improper axis means the S axis, right? We have S2, S4, S6 and all that, right? But S2 is essentially equivalent to I. S2 is I, the center of inversion. When we are here in crystals, when we are talking about rotational symmetry, we are only talking about the pure rotational axis. We do not have any impure rotational axis in crystallographic symmetry. In when we in crystals, one, two, three, four, six. Then one bar, two bar, three bar, four bar, and six bar. These 10 are the rotational axes in crystals, where 1, 2, 3, 4, 6 are the pure rotational axis, and the others are not impure, they are not improper rotational axis, they are roto inversion axis. See, in molecules, when we talk about S, that is a roto reflection axis. But here these one, 2, 3, 4, and 6, so these are roto inversion axis. This S axis we do not have in crystallographic symmetry, which are the improper axis of rotation. We do not use that notation. We only have the pure rotational axis and we have roto inversion axis. So roto inversion axis means if we have one fold, it will rotate it by 360 degree and then invert. Inversion means if we draw the stereographic projection like this, so if we have something here, then one rotate by 360 degree, it will come here, and then bar means we will invert, so this up will simply go down like this. And then for yeah, okay. No, this course is self sufficient, right, to understand. So you have covered week 1 and week 2, right? So here this 2D projection, understanding of space groups, so and the stereographic projections. So from like what I have learned is also basically from here only. So that is why. So where exactly do you need the clarity, if I may ask? See, this what I will suggest is compared to the books. So it is better to visualize to understand from YouTube than from the books, because the books were written like a time ago, a certain amount of time ago. For me personally, it is difficult to visualize from books than to visualize from YouTube. And apart from this course, so if you just type the symmetries in Google or in YouTube, you will get links where they will explain it to you, some link or the other, because I have also learned basically from the internet. Yeah. Yes, so yes, I understand, but these things, this visualization, so it is just like when we started to learn the stereochemistry. So, stereochemistry, if we learn from book, but if we use models to learn, to visualize, it is much easier if we visualize using models. Like the cyclohexane, for cyclohexane, if we try to understand the stereochemistries. Yeah, so you can search in Google, in YouTube, you will get links where they will explain it to you through animation or something. To solve. Solve means as in crystal structure. Uh, yeah, so the best thing you can do is to just crystallize a pure enantiomer. If, for example, if you have histidine in your lab, because if we look at amino acids, so glycine is not chiral. Glycine has a CH2, so glycine, yeah, achiral. But if you try to crystallize the other amino acids, so you can just start with histidine. Histidine gives nice crystals, and you can just dissolve it in water. You can heat it at 60-70 degrees, and if you then let it cool slowly, from a saturated solution, you will get nice crystals of histidine. Yeah, and it will, you can get it. Hmm. Yes, yes. Week two, there is Miller indices, right? Week two. So week two is all about Miller indices, right? Yeah, okay. Yeah, but the questions given in the assignment per week is based on that week only. So that whatever is taught, questions are made from there only. So let's see. We will see. So you go through them or through the entire video, because sometimes the questions can come from, from the middle, from anything the professor has said, which is like an important key point. Okay. Hmm. Yes, yes, yes. You go through, and if you have any question, so you have asked one question, right? Yeah. So about that P21 over M, you have asked, right, or someone else has asked? Uh, So this question, okay, okay. See, so yeah. So what was the question? So was that an assignment question? Okay. So can you tell me the number? Question number. Okay. So this I will discuss in the next live session. Precisely this question. Okay. Question 5. And you had asked, right, in this video what is happening. So this part. Yes. So here what is happening. So let me just fast forward. So yeah, yeah. So glide basically means you have a reflection and then you have a translation. So if I consider this, so this is a 9, right? This is a 9, and then this plus sign means that it is above the plane. You know this, right? Means this white page, if I consider it as one plane, then this 9 is above the plane. So that is why it is a plus. Now, if I consider this to be my glide, now can you just mute? So I am explaining. So when you have a when you can't understand, you unmute and ask. So I have this 9 and I have a plus. So it is located here. Now I will perform a glide operation on it. So now if I consider this arrow, this blue arrow, this is the direction of my b axis. This is the direction of the b axis, and perpendicular to it is the direction of my c axis. Okay, now if I want to perform the glide, then for glide I need to reflect it. For reflection, I need a mirror, and a mirror is a plane. For rotation, we need one axis about which we can rotate, but for reflection, we need a mirror like this. We need a plane so that this point can reflect here. So here we have to consider a plane along. So firstly, can you? So I am only saying. So this plane of this whiteboard. What is this plane? This plane is the BC plane. This is the B axis, this is the C axis. So this plane is my BC plane. Now the perpendicular to these two axes is the A axis. The A axis which goes through the screen and outside the A axis, and then that AC plane which is like this. This AC plane, if I consider that to be my glide plane, so what will happen? This 9 will reflect here and come here like a 6. So this 9 will reflect and come here like a 6, and the plus will remain as such. And then it will go like this. So if I draw it somewhere, I'm just drawing it. Okay. So if I have something like a 9, and if I reflect it here, it will become like this. Right? So this is exactly what is happening. Oh, I'm sorry. I'm sorry. No, no, I'm sorry. Yeah, no, no. I am sorry. I made one small mistake. So let me make it full screen. Yeah. So I had a 9 like this. If I reflect, it will be reflect like this. this is a 9, this is a 6. This is not reflection. Okay, so this curve is this side and this curve is coming this side. So if I reflect in a mirror, so this will become like this, and this plus will remain as plus because the mirror is like this. Okay, so that is exactly what is shown here. This is will go here, and then it will it will do the translation, and then it will come here. So this is the equivalent point. So what is your question? If it is 6, it is no, that depends. So if this is my 9, okay, and if I have a twofold like this, if I rotate, then it will become like this. If I perform the twofold along this direction, but if I perform twofold along this direction, then it what will it be? It will be like this. Okay, but this is not a 6. This is the 6. Excuse me, I couldn't understand. So that is clear, na? Okay. No, no, it's okay. So we were here. So about this rotational axis and the impetus. So all this has actually been explained by Sir. Okay, so what are the basic symmetries which we use in crystallography? So why we do not use a rotoreflection axis, and why we use a rotoinversion axis? What are the notations? So all this has been, the basics have been explained in the first 2 weeks. So you have to go through them, and so now if we continue from here. So this was one question. So whether S6 is equivalent to 3 bar or not. So you know what is 3 bar, right? So 3 bar is a roto inversion axis. So if I move to stereographic projections like this, so this is the point which we have now. Now, 3 bar essentially means we have a threefold followed by the inversion. So, at first, if we translate it, we will generate these points, right? This is not for 3 bar, okay? So, this is only for if we have only a threefold, like pure translation axis, we will get this 3, okay? Now, you should tell me, so what should be the stereographic projection for 3 bar? So this is only for 3. Now if I want to do for 3 bar, what should I do? Let me first delete these points. So now I want to draw the stereographic projection of 3 bar. So let me first start with the point here. Now first 3 means we will move it by 360 divided by 3, that is 120 degrees. If I move this point by 120 degrees, I will reach here, this point. Now I will perform the inversion. So this is the center. So if I perform the center of inversion, then where should I land? So the if I perform center of inversion, I will land exactly opposite to this point, which is here. Now this was up. So full circles mean that we are in the up, and these hollow circle means we are down. So if we perform the threefold on this, we will arrive here in the upward direction. Then inversion means we will land here. So this will come here. Did you get it? Am I audible or is there some problem? Um, okay, so I'm just proceeding. So we had this point. Now if we perform, if we rotate it by 120 degrees, we will come here, followed by inversion, we will move here down. Now if we take this point, so and rotate, perform the 3 bar operation here. So at first we will rotate it by 120 degrees. So rotating by 120 degrees means we will come here, then followed by inversion means the down will become up here. Now for this point, if we rotate it by 120 degrees, we will come here up, and then inversion means down will come here. And last but not the least, for this point, it is hollow circle, it is below. So if we rotate it by 120 degrees, it will come here. Then followed by inversion, it will come up here. For this one, if we perform 120 degrees, it will become where? Here. And then followed by second. If we rotate this by 120 degrees, but if we rotate this by so there is some problem here. So at first we had this. If we rotate it by 120 degrees followed by inversion, we have this 120 degrees here by inversion. This, then we rotate by 120 degrees up followed by inversion. We come here and we rotate this by 120 degrees followed by inversion. And we rotate it 120 degrees followed by inversion. Yes. 190 means there is no two. 180. Yeah, so we started. Yeah, we started here. Then first 120 is here. Then inversion. So we are coming here. So that is 1. Yes, so now this will go here. Right. Then followed by inversion. So it will, the down will become up. So this kind of thing should come here. Then this will go here. Then this should become down. Then after this, no, this is something is happening. So basically it should be like this. So I drew it before. It should be like this. So while doing, yeah, so here this point is wrong. Yeah, I'll this from we can continue from here next week. So are there any questions? Yes. You are asking about question number 5 of assignment 2. Question. No, no, you should go through the video. If you understand the video, see, all the questions will not be exactly what the professor has taught. But if you go through the videos and you understand it, you should be able to solve similar questions. So that is the idea. Okay. So you go through the videos very nicely. You should understand the video. Then you will be able to answer something which is asked similar to the video, not exactly what is in the video. Okay. 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 So any other questions? If there are no other questions, then we can stop here.